We are going to start. Our speaker today is Dr. Sue Dharmawardhane.
No, you did good.
Dr. Sue, she did her bachelor degree at the University of Colombo in Sri Lanka. Then she moved to the United States and did her PhD in the University of Massachusetts, and then did two postdocs in Albert Einstein College of Medicine in New York and University of California in San Diego. Now she is an assistant professor at the University of Texas at Austin, in the United States. And then she will move to Puerto Rico to work in the University of School of Medicine in Bayamón, UCC. We call it UCC. Today she will, another important thing. Her area of research is signal transduction in breast cancer metastasis. She has several Grammy Awards right now, and she has an excellent record of publications. Today she will talk to us about signal transduction to the actin cytoskeleton during breast cancer metastasis.
Okay, well, thank you for that, and I want to thank Dr. Edna Cora for inviting me here. And so I'm going to talk about my work on looking at the actin cytoskeleton during breast cancer metastasis. Cancer metastasis is the establishment of secondary tumors at a site other than the primary tumor. So this is a multifaceted process where you have many steps that can go wrong to promote cancer. So in cancers that start at the epithelium, these cells, the malignant cancer cells, have to move away from the primary tumor, invade the basal lamina, invade the connective tissue to get at the circulatory system. Once in the circulatory system, these cells have to evade the host immune system, extravasate through the endothelial cell layer, and find a suitable host environment to produce a secondary tumor. So all of this takes a lot of different signaling that can go wrong, and one of our ideas is to try and understand what happens during breast cancer metastasis. In breast cancer, which is a very prevalent disease in the US, they predict that in 2005, 1 in 8 women will be diagnosed with breast cancer. And the cause of death from breast cancer is usually due to metastatic cancer, because the primary cancers can be cured usually by surgery or chemotherapy. In breast cancer, the death becomes when the tumors move away to a distant site that is more vital than a breast. So in my laboratory, our broad research objective is to elucidate signaling pathways that are deregulated in breast cancer metastasis. Our research focus right now is invasion, the first step of breast cancer metastasis. And the strategy is a combination of molecular genetic, biochemical, and cell biological analysis to try and understand the signaling pathways that go wrong during invasion. I'm going to talk to you mainly about our work with the female hormone estrogen, phytoestrogens with estrogen-like compounds that are from plants and therefore are in your diet, and also a family of proteins called Rho GTPases. And the model systems I'm going to be talking about are human breast cancer cell lines in tissue culture and the nude mouse model of experimental metastasis. So invasion requires motile mechanisms, especially for breast cancer, which starts in the epithelial cells. Ductal or lobular carcinomas, the epithelial cells will undergo a mesenchymal transition and become more like fibroblasts. And fibroblast motility is well studied in normal cell lines, and we know that actin cytoskeletal changes that polarize the cells are required to make the cells move forward. I'm going to be talking about structures called Lamellipodia, and these are cell surface actin structures that have cross-linked actin filaments that push the cell forward. I'm also going to be talking about structures called focal adhesions, and these are multimolecular signaling and structural protein complexes that anchor the actin cytoskeleton to the extracellular matrix. So in a cell that's moving forward, you're going to have dynamic interactions here where you'll have more focal adhesions in the front end that are turning over dramatically, and the focal adhesions at the tail are less, and they make the cell contract forward. So Rho GTPases are a family of small GTP binding proteins. So these are proteins that are activated by binding to GTP and are inactive when bound to GDP. And in normal cells, they act as molecular switches that are activated by cell surface receptor signaling to modulate changes in the actin cytoskeleton. Done by Anne Ridley when she was in Ann Hall's laboratory in 1992, very clearly showed that different Rho GTPases can make very different actin structures. So what I'm just showing here is cells stained with rhodamine phalloidin to look at polymerized actin, and cells stained with an anti-phosphotyrosine antibody to look at focal adhesions, because focal adhesions tend to have a lot of tyrosine phosphorylated proteins. And what they showed was that Rho will cause structures called stress fibers, RAC will cause structures called Lamellipodia, which have cross-linked actin networks and different actin binding proteins that cross-link the actin. RAC also causes a lot of small focal adhesions around the Lamellipodia that are important for motility. Then CDC42, a closely related protein, makes Filopodia, which are structures made up of parallel actin bundles. So these are these thin needle-like structures that I'm going to be talking a lot about during my talk, and it also makes focal adhesions. So I will refer to a lot on RAC and CDC42 since they are known to be important for motility. RAC will make Lamellipodia that induces motility, whereas the Filopodia are considered to be more sensory structures that sense the environment and chemoattractant gradients. So I'm going to be talking about 3 projects in the laboratory, but I'll spend most of my time talking about the role of estrogen and dietary phytoestrogens in signaling to the actin cytoskeleton, where our objective is to investigate a role for estrogen and soy and red wine compounds that are related to estrogen in breast cancer progression. I'll then talk a little bit about another project in the laboratory where we're looking at directly at the Rho GTPases for a role in breast cancer metastasis, and I'll end by talking to you about some of the new directions in my research where we're trying to design modalities for intravital imaging of breast cancer. So the first part of my talk was done by my graduate student Nicolas Asios when he was a technician and now a PhD candidate in the laboratory. So I'm going to be talking mostly about estrogen and phytoestrogens. Estrogen, the female hormone, is obviously well known for its effects on sexual characteristics, development of breast and the uterus, especially during pregnancy. It's also beneficial for brain and bone strength. The harmful effects are in older women when you have estrogen activity in developed breast. So there's elevated estrogen receptor alpha levels are associated with 50 to 80% of primary breast cancers. Metastatic breast cancers tend to often lose estrogen receptor alpha expression, but they still retain the estrogen receptor beta form and also overexpress oncogenes like epidermal growth factor receptor isoforms. So we are more interested in this stage of the disease. In terms of estrogen signaling, it's very well established that estrogen acts as a steroid hormone, binds estrogen receptors that can now act as transcription factors and transcribe genes by binding to estrogen response elements in promoter regions and transcribe genes that are important for cell proliferation and therefore differentiation. The problem begins when the cells proliferate uncontrollably without going through differentiation, so you have the establishment of primary breast cancers. In terms of what we study and what we are interested in the laboratory is a more recently discovered mode of signaling where estrogen receptors not only act as gene transcription factors but are found recruited to the membrane in special caveoli-rich areas where they can cross-activate a tyrosine kinase type receptors. So what we are interested in is the activation of estrogen and phytoestrogens, the membrane estrogen receptor, that have been shown to crosstalk with epidermal growth factor receptor through non-receptor tyrosine kinases like SARC and SHIC. Many groups, including my laboratory, have shown that this signaling can activate PI3 kinase, which modulate phosphoinositol signaling to activate a protein called ACT, which is important for cell survival. But for the purpose of this talk, I want to focus on what we are looking at in terms of crosstalk with the epidermal growth factor receptor, which is well known to activate proteins like focal adhesion kinase, RAC, and CDC42, and their downstream effectors to modulate focal adhesions and the actin cytoskeleton to result in cell migration. So when we first started this project, the question we asked was, does estrogen and compounds that look like estrogen, other xenoestrogens, activate the epidermal growth factor receptor to modulate the actin cytoskeleton? So the compounds I'm going to be talking about are estrogen, the female hormone that's important for breast development, genistein and daidzein, which are isoflavones that structurally similar to estrogen and therefore can bind the estrogen receptor, that are found from soybean. And resveratrol, this is a compound, a stilbene, that is found in the grapes, in grape skins, and therefore in high quantities in red wine. And the resveratrol can also act as an estrogenic compound or an anti-estrogenic compound. So in terms of phytoestrogens in human health, phytoestrogens are important because they can bind both the estrogen receptor alpha and beta, and they can either activate genes or inactivate them. They can prevent or promote cell proliferation, be apoptotic or anti-apoptotic depending on the cell type. They can also, because of the structure, act as antioxidants and be anti-angiogenic and anti-inflammatory, so they're beneficial to cardiovascular health. They prevent osteoporosis. So there's this huge potential for phytoestrogens as alternatives for postmenopausal hormone therapy, because estrogen and progestin therapy can be detrimental to cardiovascular health as well as promote breast and uterine cancer. So when we started working on this, we knew there was a lot of data on the role of phytoestrogens in cancer prevention in terms of preventing initiation. What we wanted to know was, does it also prevent or promote cancer metastasis? So in terms of breast cancer, we want to understand the effectiveness of these dietary phytoestrogens in breast cancer patients, survivors, or women at high risk for breast cancer. So we used a simple strategy by looking at the cell biology first, and we wanted to look at the role for estrogen and phytoestrogens in cell migration, focal adhesion turnover, and focal adhesion kinase activity, because focal adhesion kinase is known to modulate focal adhesion turnover and be important for invasion. And then we also looked at actin cytoskeletal rearrangement, and the model system I'm going to be talking about are the human breast cancer cell lines at different stages of the disease, so they have different estrogen receptor and epidermal growth factor receptor isoform expression. So one of the first questions we asked was, does estrogen and soy phytoestrogens, genistein, that I'm going to be showing as G, and daidzein as D, so this is 50 and 100 micromolar. So what we did was, to ask this question, we used a transwell motility assay, where the chemoattractant here is either the vehicle, estrogen, EGF, genistein, or daidzein. Then we place quiescent cells on top of this inner well, and the membrane here has 8 micron diameter pores that will enable the cells to migrate through by an active migratory mechanism. So 8 hours later, we come and count the number of cells that came through to this side, and what you see here is relative number of cells per field. But the bottom line here is that we found, now epidermal growth factor was our positive control. We found that estrogen, genistein, and daidzein acted similar to EGF and promoted cell migration. And these cells we used were the ER alpha minus beta plus metastatic cell line MDA MB231. So then we asked the question, is this increase in cell migration due to a structural difference that these compounds are promoting. So we looked at focal adhesions by staining with an anti-phosphotyrosine antibody. So the green foci you see here are focal adhesions. So epidermal growth factor was our positive control. So you see lots of small focal adhesions. Similarly, estrogen, genistein, and daidzein also gave a lot of focal adhesions in these cells. And this is just counting. I made Nico count 100, at least 100 cells per treatment, and we count the foci by hand. And what you see here is that EGF-E2 and genistein and daidzein at different concentrations increase focal adhesions in this cell line. So then we wanted to know if there was a mechanistic reason for this. And we looked at focal adhesion kinase activity because it's one of the first proteins that gets recruited to focal adhesions when integrin receptors are activated to dynamically modulate the focal adhesions. So what you see here are examples of representative Western blots that we use for our scanning densitometry that's shown in these graphs. These are whole cell lysates, vehicle, estrogen, genistein at different concentrations, and then we stain with a anti-phospho-FAC antibody which detects only the active form of FAC. So this is just total FAC here. So what we found was that estrogen and EGF increased phospho-FAC as expected. And then the genistein and daidzein at 3 different concentrations here, but we've done concentration curves for both of these compounds in ER alpha, beta plus T47D human breast cancer cell lines, as well as the ER alpha minus beta plus MDA MB231, and we see the same, that estrogen, genistein, and daidzein activate FAC in a concentration dependent manner. Therefore, we think that the focal adhesion increase may be due to focal adhesion kinase activity increase. So then we wanted to look at the actin cytoskeleton. So these are fluorescence micrographs stained with a rhodamine tagged phalloidin that we are looking for polymerized actin specifically. So you can see our positive control EGF makes the structures called Lamellipodia that are important for cell migration. Estrogen, genistein, and daidzein acted similar to EGF by making Lamellipodia. So this could be the reason why these cells are more motile in response to these compounds. So from a lot of work done with normal cell lines, we know that the small GTP binding protein RAC regulates Lamellipodia formation during migration. So what we did was to take our ER alpha minus beta plus MB231 and express a dominant active form of RAC in it to test to see if the same pathways are working in this cell line. And yes, you see the cross-linked actin organized into Lamellipodia, and this cell is going to move this way when the RAC protein is dominant active. So this particular amino acid substitution in the GTP binding domain would render always constitutively active protein. So then to answer our question, if RAC was what was being activated by estrogen or phytoestrogens during Lamellipodia formation, what we did was to do the other experiment where we expressed a dominant negative form of RAC. So what you see here are the vector alone controls in dark blue, and in light blue are the Lamellipodia number per cell for cells that stably expressed RAC N17, which is a dominant negative construct. So what we found was that EGF and E2, genistein and daidzein increase Lamellipodia per cell in the control cell line, but in the cell line that stably expressed a dominant negative RAC, we were able to block the effect for all compounds, indicating that these compounds were activating Lamellipodia formation through RAC. So just to recap what I've been saying, this is our working hypothesis, and we need to do more work on the receptors here. But genistein, daidzein, and estrogen may activate the membrane estrogen receptors to activate epidermal growth factor receptor signaling, to activate FAC and RAC, and therefore modulate the actin cytoskeleton to make structures that will induce forward migration. So we were interested, when we went into this project, we really wanted to look for a breast cancer metastasis preventive, and these compounds look like they are activating breast cancer. So we decided to look at a well-known cancer preventive, resveratrol, which is considered to be really important for cardiovascular health and therefore to be the beneficial effects of the Mediterranean diet. So we wanted to look at resveratrol in the context of breast cancer prevention in already established breast cancer. So we are again using the same model where we have the ER alpha minus, beta plus, EGFR plus cell line. And in similar motility assays, EGF and estrogen are positive controls and they increase migration. Resveratrol decreased migration. So we found this a very interesting result, and we wanted to then again go back and do similar experiments that I showed you before and try to see if there was a structural and a mechanistic reason for why resveratrol was decreased in cell migration. Resveratrol does not induce apoptosis in these cells at the time that we did these assays. So we looked at focal adhesions again by staining for phosphotyrosine residues. So this green foci, if you count the number of focal adhesions per cell for at least 100 cells per treatment, what we saw was that again our positive controls were estrogen and EGF. Interestingly, resveratrol reduced the number of focal adhesions. This is in 10 minutes. And if you add resveratrol for 10 minutes and then added estrogen for 10 minutes, we were able to get the resveratrol effect, indicating that resveratrol was effectively competing with estrogen for the estrogen receptor. However, if you added resveratrol for 10 minutes and then added EGF, we could not inhibit focal adhesion formation probably because EGF is a better ligand for its own receptor. So then we pursued this further by using tiafostan AG1478, which is a specific EGFR1 inhibitor. So if you look at the number of focal adhesions per cell minus and plus AG1478, we found that this inhibitor was able to inhibit the number of focal adhesions induced in response to estrogen as well as EGF. Resveratrol, we don't see a difference because resveratrol is already inhibiting it. So we think that estrogen signaling through epidermal growth factor receptor will induce focal adhesions, and that resveratrol acts different to estrogen by reducing the number of focal adhesions. So then we wanted to look at focal adhesion kinase activity. So we did similar experiments that I showed you before, where you Western blot whole cell lysates that have been treated with resveratrol at different concentrations for 10 minutes, and then look at the ability of FACT to be activated by Western blotting with a specific anti-phospho-FACT antibody. So for both cell lines, the T47D ER alpha minus beta plus and MB231 ER alpha minus beta plus cell line, we found that resveratrol decreased FACT activity in a concentration dependent manner. So this could be the reason why resveratrol is decreasing focal adhesions. So then we looked at actin cytoskeletal structures, and as I showed you before, estradiol and EGF makes Lamellipodia in 10 minutes, and these are the cells that are polarized and induce motility. With resveratrol, we saw something very interesting. Resveratrol induced this global array of Filopodia, the thin needle-like projections of actin that have different actin binding protein complement. And these cells were also not polarized, similar to estrogen and EGF treated cells. So this was a concentration dependent effect, and we started using resveratrol at 50 micromolar because that gives us the best effect. So then we wanted to know if estrogen receptor was important for resveratrol's effect on the actin cytoskeleton. To do that, we used a panel of cell lines. T47D is an estrogen receptor alpha and beta plus cell line, and you can see that estrogen makes Lamellipodia and resveratrol induces Filopodia in this cell line. If you look at MB231, the ER alpha minus ER beta plus cell line, estrogen makes Lamellipodia, resveratrol makes Filopodia. However, when we looked at SKBR3, a cell line that had lost both ER alpha and beta expression, you don't see any actin cytoskeletal changes in response to estrogen or resveratrol. Therefore, we think there is at least a dependence on estrogen receptor beta for modulation of the actin cytoskeleton by resveratrol or estrogen. So right now we are doing the right experiment to confirm this, where we will express ectopically the ER beta or ER alpha and see if we can recapitulate the resveratrol or estrogen effect on the actin cytoskeleton. So to understand how Filopodia formation is regulating migration, we've done more experiments, and what I'm showing you here is that the effect of resveratrol on cell migration seems to have an inverse relationship with the effect of resveratrol on the actin cytoskeleton. So in terms of cell migration, resveratrol inhibits cell migration, whereas estrogen and EGF will increase cell migration. If you add resveratrol for 10 minutes and then treat with estrogen for 8 hours, you still should see decreased migration. If you treat the cells with resveratrol for 10 minutes and then EGF for 8 hours, you still decrease cell migration, which is an interesting result because that means resveratrol can act as an inhibitor of cell migration in physiological conditions. With the Filopodia effect, estrogen and EGF do not induce Filopodia, resveratrol does. And if you add resveratrol before adding estrogen, you can still have the Filopodia effect. So resveratrol effectively competes with estrogen for the estrogen receptor, but it does not compete with EGF for the EGF receptor. So to look at this further, we looked at the role of adding the AG1478, the EGFR1 inhibitor, on EGFR activity in response to estrogen, EGF, or positive control, or resveratrol, and we also looked at Filopodia extension. So what I'm showing you here is that these are total cell lysates, western blotted, with an anti-EGFR or an anti-phospho-EGFR that looks at activated form of the EGFR. And you can see that with estrogen, we do see EGFR activity that can be inhibited by addition of the AG1478 inhibitor. With EGF, our positive control, we see a lot of activity that can be inhibited. Interestingly, with resveratrol, we saw an increase in EGFR activity that could not be inhibited by adding this inhibitor. So we think that resveratrol may signal via EGFR, but by a different signal pathway than what you would see when you activate the receptor with EGF or estrogen crosstalk. So this is, there's precedence in the literature for AG1478 insensitive signaling to the EGFR by SARC. So we think that resveratrol may be modulating estrogen receptor activity to activate SARC and therefore activate EGFR in a different manner from EGFR-E2. If you look at Filopodia extension, interestingly, when we added the AG1478 and looked at the number of Filopodia per cell, the resveratrol treated cells did induce Filopodia, but to a lesser extent than in the absence of the inhibitor. So we think that it is at least partially regulated by EGFR activity. So then we wanted to look a little bit more at downstream signaling and decided to look at the Rho GTPases. CDC42, I first showed you, was important for making Filopodia. CDC42 signal into proteins like WASP will recruit actin and polymerize actin to make these thin needle-like projections that are sensory structures. And CDC42 also can polarize cells. CDC42 can activate RAC, but RAC, the related GTPase, can also be activated by EGF to activate different downstream effectors to make Lamellipodia that are useful for cell motility. So we wanted to look at the role of resveratrol signaling to CDC42 or RAC to see if that was how the actin cytoskeleton was being changed. So to answer that question, what Nico did was to go and make a stable cell line in the MDA-MB231 background that now expressed a MIG-tagged CDC42T17N, which is an amino acid substitution that makes a dominant negative protein. So what you see here are the control cells, And here the positive control we used was bradykinin. It's a cytokine that's known to directly signal to CDC42 and induce Filopodia. So you can see that in control cells, both bradykinin and resveratrol induce Filopodia. In the CDC42 T17N expressing cells, the bradykinin-induced Filopodia response was completely inhibited as expected, but resveratrol-induced Filopodia was still, it was partially inhibited but not completely inhibited. So then we went and looked at endogenous CDC42 activity to see if resveratrol was directly activating CDC42. So what you see here is a pull-down assay where we utilize a downstream effector of CDC42. It's a GST fusion protein with the binding domain of this effector that only binds the GTP bound active form of CDC42. So in this pull-down assay, this is our positive control, and here are the active CDC42 that we detected in vehicle-treated cells, EGF-treated cells, resveratrol-treated cells, or cells treated with resveratrol first and then EGF. And in any of these, we really didn't see a significant increase in CDC42 activity. So resveratrol does not seem to alter endogenous CDC42 activity. So because of that, we decided to look at RAC, a related protein that we know is important for modulating Lamellipodia formation. So here's the RAC T17N stably expressed in cell line. So this is the MIC tag. And with the control cells, again, we have the expected result. If you look at the relative number of Filopodia per cell, resveratrol increases Filopodia. And over here are the cells that express the RAC1 dominant negative construct, and they also act similar and express Filopodia in response to resveratrol, but not EGFR E2. So we never saw a blocking of the Filopodia response to resveratrol by blocking RAC activity. What was interesting is when we looked at the ability of resveratrol to activate endogenous RAC by doing the pull-down assay that I just described, we found that EGF, our positive control, activated RAC, but resveratrol activated RAC even when we added resveratrol before EGF. So this is the quantification of 3 experiments like this, and very clearly resveratrol at 10 minutes seems to activate RAC. So just to correlate what I'm talking about in terms of Filopodia formation, because we started doing these experiments to say that the resveratrol-induced Filopodia response was modulated by Rho GTPases. And we used classic RAC and CDC42, which are classically known to activate active cytoskeletal dynamics. So what we found here is if you look at the time curve of Filopodia response, resveratrol at 50 micromolar will induce Filopodia starting at 2 minutes, peak at 10 minutes, and then this response is sustained for 60 minutes. If you look at CDC42 activity, we don't see a whole lot of difference. If you look at resveratrol activity with time, we do see an increase at 10 minutes, but this is down to basal levels by 60 minutes. So what this is showing is that RAC activity alone is insufficient to induce the Filopodia and sustain the Filopodia response to resveratrol. So right now we are looking at other more recently identified Rho GTPases that may have a role in addition to RAC and CDC42 in resveratrol-induced Filopodia extension. So just to conclude this section, what I've been telling you so far is that estrogen and soy phytoestrogens increase cell survival, cell migration, focal adhesion kinase activity, and focal adhesion number. And we also know that estrogen and soy phytoestrogens induce leading edge Lamellipodia via RAC signaling to induce cell migration, and therefore may have more of a promotive role in breast cancer metastasis. Resveratrol acts opposite by decreasing cell survival, cell migration, FAC activity, and focal adhesion number. And resveratrol induces a global and sustained Filopodia response that may decrease cell migration and therefore act as a preventive in breast cancer metastasis. So this is like our working hypothesis right now. I showed you that soy phytoestrogens and estradiol can cross-activate the EGF receptor to activate FAC, RAC, and Lamellipodia and induce cell migration. We think resveratrol does go through the same pathway, but by differential signaling, either via RAC and CDC42 and a recently identified protein called RIF that belongs to the same family that can activate Filopodia by in a kind of a co-stimulatory manner with RAC, and we are looking at that right now. It's also possible that resveratrol may induce direct downstream effectors of RAC and CDC42 that will involve Filopodia activation through other adapter proteins or modulation of membrane phospholipids. So these are all potential pathways that we are interested in pursuing in the laboratory. But ultimately the idea is that resveratrol will inhibit cell invasion and therefore act as a preventive. So our future directions are, we just got funded to look at the rapid signaling pathways that are mediated upstream, and we also have a project we are going to start where the idea is to look at this in vivo, where we would establish mammary tumors in mice, then feed them estrogen, genistein, daidzein, or resveratrol in their diet and check to see if that's going to induce or prevent promotion of breast cancer. And we are going to do this by live image analysis, so I'm going to talk to you about that at the end of my talk. So I'm going to take a few minutes, not too long, it's not such a long story, just to tell you what we are doing with looking at RAC isoforms as direct stimulators of breast cancer metastasis. And then I'll end by talking to you about the novel imaging modalities that we are analyzing in the laboratory. So this work was done by my graduate students Paige Boka and Lakshmi Krishnamurthy. Paige just graduated with this work, and the objective here is to establish a role for RAC1 and RAC3, a close isoform of RAC1 in breast cancer metastasis. So the model system we are using here are metastatic variant of a human high metastatic breast cancer cell line, MDA-MB435. Our collaborator, Dr. Janet Price at the MD Anderson Cancer Center in Houston, took this one cell line and she cycled it through a mouse model and isolated different metastatic variants based on their ability to induce metastasis. So we have high to low metastatic variants of the same cell line, and the idea here is that they would have the same genetic background but be different in the metastasis promoters and suppressors. Then we correlated, using these variants, we were able to correlate high metastatic efficiency with a more migratory and invasive phenotype. I don't have time to show you the data, but Paige also showed that the high metastatic variants had more RAC1 and RAC3 endogenous activity. So then the strategy we used was to take this metastatic variant and look at the effect of expressing dominant negative or dominant active RAC1 or RAC3 in vitro or in vivo. So what I'm showing you is like an abbreviated form of all the data we have where we analyze these mutants in vitro, and right here what I'm showing you is the vector alone control of these metastatic variant expressing RAC1 or RAC3 dominant active proteins. And what this is showing is that you can activate just RAC1 or RAC3 and make the least metastatic variant act more like a high metastatic variant. If you take the high metastatic variant and look at the effects on cell adhesion, migration, or invasion, it's very high. But if you take the cell line now and stably express a dominant negative RAC1 or a RAC3, we can block these metastatic activities. So in vitro, it looks like RAC1 or RAC3 can affect breast cancer metastasis. So then we wanted to see if we could also get this to work in vivo, and we went to the nude mouse model where we use immunocompromised athymic nude mice and inject the human breast cancer cell lines into their mammary fat pad to induce primary tumors. So then about 2 months later, what we do is to sacrifice the animals infuse the lung with India ink, then bleach them. So what you see in white are the metastases. So then you just count the number of lung metastases per primary tumor. I don't have all the data analyzed to show you, but these are preliminary studies that show that if you have the least metastatic cell variant and established primary tumors, remember this is the same background, so we have no problem with establishing primary tumors with this cell line. And then you look at the number of lung metastases, this cell line does not metastasize to the lung. But if you express a dominant active RAC1 or RAC3, you increase the number of metastases per lung. Similarly, if you take the most metastatic variant, we see a lot of lung metastases, and if you express a dominant negative RAC1 or a RAC3, you do not see any lung metastases. So we think that both in vitro and in vivo, we've shown a role for RAC1 and RAC3 in breast cancer metastasis. So the conclusions from this section is that we have established a positive correlation between metastatic efficiency and a migratory phenotype, as well as high endogenous RAC activity. So if you stably express a dominant active RAC, you can recapitulate the more metastatic phenotype, and stable expression of a dominant negative RAC can block that metastatic phenotype. So our future directions here is because we found that the high metastatic variants have high RAC activity, we think that the upstream effectors of RAC are being activated in these cell lines. So we want to do immunoprecipitates of the dominant negative construct to see if we have more upstream effectors that are being associated with these immunoprecipitates, and do a proteomic analysis to identify the effectors that are different in the metastatic variants. We also want to do an in vivo analysis of fluorescently tagged breast cancers that express the dominant active or dominant negative Rho GTPases to do a better mutant analysis. Because the way we do our metastasis assays, we only detect the first stage, the primary tumor, and then the lung metastasis. But metastasis is a multifaceted process that if we are interested in the first step, the invasion, we don't really see it the way we analyze our data. So what I wanted to do was to try and develop methods that will detect fluorescently tagged breast cancer cells leaving the primary tumor. To do that, I contacted Dr. Rebecca Richards-Cordham in biomedical engineering, where she has a lot of experience with designing methods for in situ cancer detection. So she and I co-advised these two students, Michelle Hoffman and Alicia Lacey, who are developing methods for non-invasive analysis of breast cancer in mouse models where the mice have been injected with fluorescent protein tagged breast cancer cell lines. So we use the red fluorescent protein from jellyfish, we use the cDNA to stably integrate into our cells to make red cells. And the instrument that I'm going to show you very quickly is a macroscopic illumination system that's very easily developed by Alicia Lacey, who's a biomedical engineer. And basically it has a xenon lamp source that goes through an excitation filter wheel that we can control. So we can control either white light, green light, or blue light that goes through a fiber optic light guide and comes through an excitation probe. So we can use any fluorescent protein tagged biological system right here. And what you're seeing is this little mouse that's just been held by someone. We don't use anesthesia, we do not use surgery. So this is completely non-invasive, and we can follow this in the same mouse by just exciting the fluorescent protein tagged breast cancers and then detecting the emission with a filter that's attached directly to a digital camera, so we can analyze the data in a computer. So I'm just going to show you a couple of slides to show you what the data looks like, if I can get it to move. So what this shows is, I don't have time to show you the work we've done with inflammatory breast cancer, but this is a highly invasive, locally invasive form of breast cancer, and we have a cell line in the laboratory that we have expressed the red fluorescent protein. So this is what the cells look like, a pellet in the in a tube, and we can see the cells being injected under the skin of the mouse from the time of injection. So you can use this system to detect primary tumor take, primary tumors and invasion. So here's a skin mouse that we used for our imaging. We can see shadows of distant metastases, but it's really difficult to analyze the metastases directly using this system. And here's just another example of the NDA-MB435 high metastatic variant that I talked about earlier, that's tagged with red fluorescent protein, so you detect it. And in 2 months, we can also see the angiogenic vessels here. Again, like I told you, we could see where the distant metastases were, but to really analyze the metastases, we had to remove the lung. And then when you image the lung, you can really see where all the metastatic cell lines are. So we don't have to do histopathology. We can just use our system to directly monitor metastases. So the quantifications look like this, where you can start quantifying through pixel intensity breast cancer progression in any cell line and in the same mouse. Right now we are trying to develop this system to analyze the effect of chemotherapeutics and chemopreventives on breast cancer progression, and that sort of takes me back to what we were looking at with the phytoestrogens, because we can now make red fluorescent protein tag breast tumors in the mice, then give them these compounds and analyze the effect on breast cancer progression. We are also developing a confocal fluorescence system for further analysis of individual cells that leave the tumor. This is just reflectance microscopy, so we had to surgically open the mouse up, but this is still a live image. And the idea right now, Alicia Lacey is making this fluorescence capable, so we can look at fluorescence breast tumors and fluorescently tagged cells leaving the tumor. So I'm going to stop there and just tell you that yes, we have developed the macroscopic system that is being used currently to monitor the progression of fluorescent breast tumors, and we are going to further analyze this by the fluorescence confocal microscope that's been developed right now. So my whole talk, I'm really already done, is just to remind you, I talked about how estrogen and soy phytoestrogens signal to the actin cytoskeleton to induce structures called Lamellipodia and therefore promote breast cancer cell migration and maybe metastasis. I showed you that the red wine phytoestrogen resveratrol acts different by making a global and sustained array of Filopodia, which we think inhibits migration and therefore potentially act as a breast cancer metastasis preventive. RAC1 and RAC3 signal to the actin cytoskeleton to promote metastasis of breast cancer cells, and we hope that some of these new methods we are developing in terms of intravital imaging will allow us to confirm these studies in vivo and really show a role for estrogen, phytoestrogens, and Rho GTPases in breast cancer metastasis in live animal models. So I just want to end by thanking the people who actually did the work. Nicolas Asios did all of the work that I talked about with the estrogen and phytoestrogen signaling. Paige Boka and Lakshmi Krishnamurthy worked on the RAC1 and RAC3 project. All of the Mishila and Alicia are graduate students. Kristen Wall is an undergraduate student, and Vivian Mack is a technician that I share with Dr. Rebecca Riches-Cordham, who's been a wonderful collaborator. And I also want to thank my other collaborators and the people who fund our research. Any questions?
Yes. Does the resveratrol effect require internalization with the receptor, or is it just binding at the membrane level?
We don't know that right now. We want to try and tag the resveratrol and see if it binds at the membrane level or it gets internalized. Right now, we don't know. It should get internalized just because it's a small steroidal compound, but we predict that it must activate a membrane receptor in order to crosstalk with the epidermal growth factor receptor that's at the membrane. Yeah?
If the results with the insane, after when you treated the cell with 100 micromolar sane, the activity of the focal adhesion kinase looks like was
100 micromolar is cytotoxic.
Oh, okay, that's why. Okay.
Any more questions?
I just want to learn the difference between Filopodia and Lamellipodia, what their role in terms of the cell motility.
So Lamellipodia are cross-linked, cross-hatched actin filaments that usually involve binding of filament at the cross-links. So it's signal different, and it's supposed to push the actin cytoskeleton forward in such a way that the cells move forward. Whereas the Filopodia are considered to be more sensory structures, so they have parallel actin bundles and proteins like vilin and fibrin bundling the actin bundles, and they're supposed to sense the chemoattractant gradient and not be very useful for motility.
So therefore, when you add your agent, the resveratrol, so that holds the cells very tightly to the matrix, and that's why it's not able to.
It doesn't hold the cells tightly to the matrix because resveratrol also signals to FAC to reduce focal adhesions. But I think that the cells don't have a sense of direction because it's a global response and they just sit there.
Yeah, that leads to another question. Those epithelial cells are highly polarized.
The epithelial cells, yes, but the cell line we use has already gone through the epithelial to mesenchymal transition. They're fibroblast-like cells.
Okay, so but still the direction is only one direction, and they can go in either direction?
They can go in either direction as long as they sense the chemoattractive gradient that's coming from.
That's why they pass through that difference.
Yeah, exactly. Yeah.
Yes, I'm curious about what are the levels of the concentrations of these compounds and steroids that you're using compared to what you would get from the diet?
They are pretty high. In a vegetarian diet, you can get similar soy levels. And with the resveratrol, we calculated, depending on the variety of grape and the wine that you use, like a Pinot Noir, about 2 glasses of wine would have 50 micromolar resveratrol.
I'll comment on that one too, that a lot of that on the nutrition picture, that now they're recommending that soy is good, but not in huge amounts.
Exactly.
It's beginning to go into a lot of products, and there may be some risk.
Misconceptions, yeah.
Rack 1 and Rack 3, are they localized at the leading edge of the migrating cells?
Yes. Yes, they are, the active form.
I have one more question.
Mhm,
In terms
sure.
of the EGF receptor,
Uh-huh.
you're using phosphotyrosine antibody, I mean EGF specific, but
Yeah.
when you treat them, is the phosphorylation pattern differs? I don't know how many phosphorylation sites.
We only looked at tyrosine 1173, and it gets phosphorylated in many different residues, but we haven't really looked at all of those and the
So
effect
may
that,
may
yeah,
have the other phosphorylation
absolutely,
site may have.
yeah.
If no, thank you.
Well, thank you.